Hi everyone. Today we have a special guest. He's Dr. Peter Toogood, Professor of Medicinal Chemistry, University of Michigan. He's going to be explaining to us some work in epidermal growth factor receptors. And yes, and take it away, Peter. Thank you for being with us.
It's my pleasure to be here. We're going to do a short video today looking at epidermal growth factor receptor, and that's a kinase. It's a receptor tyrosine kinase, and so we're going to be looking at small molecule inhibitors of that kinase protein. We're going to look at a couple of different ones, and these are both compounds that are currently approved as drugs because epidermal growth factor receptor is overexpressed in a variety of cancers. So we're going to look at two compounds that are actually on the market and that are sold for the treatment of cancer. That's Gefitinib, sold as Iressa for the treatment of non-small cell lung carcinoma, and Lapatinib, which is sold as Tyrosine for the treatment of breast carcinoma. And so there are a few different ways that we can import those proteins into Nanome, and I'm going to use two different ways just to illustrate the point. And the first way we're going to use is just to go to the web browser that's here on my wrist, and we'll click on that, and that should bring up there we go. That should bring up a web page, and within this web page that's here in front of me, there's already a shortcut to the protein structural database, or CSB. And so we're going to click on that, and we'll go ahead and type in the PDB code 2ITY of the protein that we're going to look at first. So let me click on that and hit search. And we'll come up with a description of that protein. So this is the crystal structure of, crystal structure of EGFR kinase domain in complex with Iressa, which is Gefitinib. And we can go over here to the download files and click on the PDB format, and that will go ahead and download. And you can see this red circle that's shown up by the arrow pointing down on the top of the tab here, and there's our file. So we can click on that, and that should download that right into Nanome for us. So there we go. There's our protein, and we can grab that and bring that down so that we can take a look at it. And there we go. So this is the sort of the default way that the protein comes up. And what I like to do, one of the first things I like to do, and you know what, this is a long way away, so the first thing I like to do is select the ligand and just give it a different color. So let's color that red so we can actually see the ligand. There we go. So there's our ligand, and we can look at the structure of this ligand, not just in 3 dimensions like this, but we can actually go over to the ligand tab in our entry list and just pull out the structure. So this is the structure. Let me get the protein out of the way here for a second. Here's the structure of the Gefitinib, which is the ligand that's bound into this protein crystal structure. And you can see that it has an aminoquinazoline core, which is pretty common to the class of EGFR inhibitors that we're going to be looking at. So that's a nice way. So as I said, the aminoquinazoline core here is going to be common to both the ligands that we're looking at, and then it has an aryl substituent with a couple of simple substituents on the right-hand side corner here, and then a solubilizing chain that's coming out. And those two regions are going to differ between the two molecules that we're going to look at. Okay, so let's get back to the protein here. One of the things that I like to do is just change the way that it comes in by default. So what we'll do is let's put on all the full amino acid sequence of the protein in stick format, and we'll turn off the ribbons for the moment. And so here's our protein in stick format. And you can see where it was helpful to color the ligand because it really stands out relative to the protein here. And we can sort of manipulate this and have a look at it. And one of the things that I like to highlight in kinase structures like this is what's called the Dfg motif. And I'm going to select that for you. Let me come in here and drag it a little closer.
Yeah.
So here's a aspartate or a D, and a phenylalanine or an F, and the glycine. So there's a Dfg motif. And let me color that so it stands out. Okay, so now we've colored it green. So this is a common motif in kinases, the Dfg motif, that is indicative of the binding mode of the inhibitor. And
Yes.
in this particular case, what we can see is that the aspartate residue, the D residue, I'll just point to it right here with my finger, is pointing in towards the ligand. And the ligand is binding in the ATP pocket. So that aspartate's actually pointing into the ATP binding site. And so this conformation of the Dfg motif is what we refer to as a Dfg in conformation. And this is indicative of an active conformation of the kinase. And so that suggests to us that this ligand, gefitinib, is actually binding in a Dfg or type 1 inhibitor mode. It's binding to the active form of the kinase. And you'll see in the second protein that EGFR structure that we look at, that the Dfg motif is similarly in this Dfg in configuration or conformation. So that's an important motif to recognize in kinase crystal structures. Another thing that I like to do is put on a whole surface, and let's make it an opaque surface. Here's a protein surface that gives us an idea of the protein in 3 dimensions. And also, this is a nice way to look at the volume or the space in the pocket that the ligand is binding to. And we can color this. Let's color that surface with a slightly different color scheme to make it stand apart from the ligand. I'm looking for the YRB hydrophobic surface color pattern here. There we go. And now what we can do is we can sort of walk right into the active site of the protein where the ligand is binding, and let me unselect the ligand here.
You can click on the sky if you point your laser directly upwards and then click.
I'm so far inside the protein, I can't get to the sky. There we go.
Yeah.
So now we can see there's a water molecule down here. And what I meant to do, but I will go back and do in a second, is show that the ligand actually makes a hydrogen bond to that water molecule, as well as making a hydrogen bond to the hinge of the protein. But look, we can look at sort of the cavities in this pocket. There's a chlorine here that's taking up into a cavity at the back of the pocket. There's also a lot of space back here that is not occupied by any of the ligand. Behind the ligand here, there's a fair amount of empty space that suggests that it might be possible to design a ligand that would occupy more of the ATP binding site, perhaps to get additional potency. So this is the kind of thing that's very difficult to see in 2 dimensions on a screen, or even a 3-dimensional projection on a screen. But once you really get inside the ATP binding site of a protein like this, you really get a sense of which parts of the protein are still not interacting with the ligand and which parts of the protein are interacting with the ligand. And I think this can be a really informative way of looking at ligands and thinking about ligand design and ligand optimization when you're doing drug discovery. So let's get out of the cave.
Right. Got a Plato here, yeah. No.
Yeah. And we'll turn the surface off. Let's turn that surface off. And I mentioned something about showing the hydrogen bonds. So let's go ahead and do that. And we can come over here. We have to select the ligand, and we'll come over here and we'll use the calculate hydrogen bonds feature. And now you can see that what I was talking about when we were in the three-dimensional structure, the surface structure there's a hydrogen bond that forms between one of the nitrogens of the quinazoline ring and a backbone NH of the protein. And this is part of the so-called hinge, which connects the C-terminal lobe and the N-terminal lobe of the kinase. And then there's another hydrogen bond to a water molecule back here. And one could imagine that if you could build off of the core of the ligand in a way that would displace that water molecule, you might get some additional binding affinity as a consequence of the increased entropy of displacing that water molecule. Okay, so that's the EGFR bound to gefitinib. And again, let's just change the display here. I'm going to go ahead and put the cartoon ribbons on, and we'll take off the sticks. And I want to color the cartoon coils here. We'll just make them one solid color. Let's make them blue. And you know what? We'll also, I'm going to slightly differently color the ligand to match. So we'll make that this sort of I don't know what you call that. Maybe that's taupe. That's a bluish color, gray-blue color. So that we matched the color of the ligand with the color of the coils, and that will be helpful to us in a little bit when we superimpose a couple of different structures. Okay, so that's the 2ITY structure. Now let's go get a different structure, which is also EGFR, but bound to a different small molecule kinase inhibitor. So I'm going to go back to the load menu, and we'll pick the RCSB PDB file. And the structure I want this time is 1XKK. And this is as I said, this is a second EGFR structure. So we should recognize some similarities in this structure, but it's bound to a different ligand. So let me get that one out of the way. Now this comes in, interestingly, this comes in an orientation that makes it actually a little bit tricky to find the ligand. So let's do that same trick, and let's color the ligand some nice bright color so we can find it. And there it is. It's in red here. And we'll just sort of twist it around and orient it similar to the orientation we were looking at the first protein, the 2ITY protein. And so here's our ligand. And if we pull up the two-dimensional image again, you can see that has a similar aminoquinazoline core, which I pointed out earlier with gefitinib. So this is Lapatinib. There's an additional extension of this benzyl ether on the aniline group to the top right. And then the side chain here is a little different. Perhaps this is also similar to the solubilizing group we saw on gefitinib. This is projecting out towards the solvent, and we can see that right here. And there's an extraneous sulfate residue here that was part of the crystallization. So let's go through the same sequence here of illustrating different parts of this protein. So we'll go ahead and put the sticks on. We'll turn the ribbons off, and this will allow us to immediately find the Dfg motif there. And we'll make that green like we did before. So as I was pointing out, we actually see the same conformation of the Dfg motif, where the D is pointing in towards the ATP pocket and the phenylalanine is pointing away. And there is a different conformation of that Dfg motif that is observed in some ligand protein structures, where the aspartate residue and the phenylalanine actually swap positions as a flip of the Dfg motif, and the phenylalanine points into the ATP binding pocket. And that's referred to as a type 2 binding mode, and that's an inactive conformation of the protein. So here it looks like we've got a type 1 conformation of the protein, and we're going to look at that in a little bit more detail. It's actually not exactly the same when we get into it, but you can see that it does have the same conformation of that Dfg motif. Now let's put the hydrogen bonds on again. That was interesting when we did it before, and you can see we pick up the same hydrogen bond between the quinazoline and the backbone NH of the hinge. And there's actually a similar hydrogen bond to a water molecule in the back of the pocket. Notice there's a second water molecule here as well that doesn't appear to be interacting with the ligand at all. And then there's a water molecule in the front that is interacting with the ligand that we didn't see in the gefitinib structure. So that's some interesting similarities and differences to gefitinib. Let's go ahead and put the surface on. And again, we'll make it relatively opaque, and let's color the surface. Similarly, let's give it the similar sort of YRB hydrophobic color scheme, where the yellow represents a hydrophobic surface, and then we see blue for a basic surface and red for an acidic surface. So let's go walk into the cave again and have a look around.
Yeah, scale it up. Nice.
We go right in there, and you can see that the pocket is sort of opened up a little bit in the back there. So there's more space around, not just where that additional substituent is, that benzyl ether, which seems to have found some additional space to occupy in the back of the pocket, but also where that additional water molecule is at the back there, like all the way in here. There's almost a channel, it seems, through there. So that it seems that just getting in, looking around, that there's a larger volume, there's a bigger pocket that's available to this slightly larger ligand, suggesting that it somehow induced a change in the protein to accommodate a larger ligand. And again, you can see sort of where there are close interactions between the ligand and the protein, and where there's potentially additional space that might be occupied by additional substitution on the ligand. So I think this is a really neat way of looking at protein ligand structures, and obviously the two that we're looking at are already reasonably optimized for their targets. But if you were starting off earlier in a to lead or lead optimization campaign, and you were able to look at a structure like this, you would really be able to get some insight, I think, into where to place substituents. You can look at residues on the protein that are proximal to the ligand that you might be able to hydrogen bond to, or make additional van der Waals contacts to. So this is just, I think, so powerful for ligand design. And it's colorful. I like that too. Okay, so let's go ahead and we'll turn off that surface again, and we'll put back on the coils, and let's turn off the sticks, and I'm going to color those coils again. We'll color them, let's color them a nice yellow color. So now let's just put these two protein structures side by side. They should look pretty similar, and they do. Bear in mind that both of these are EGFR kinase domain, in fact, with a ligand bound. And so they look pretty similar to first blush, and we could kind of grab one and try to overlay it on the other, but there's an easier way to do that. And we're going to use a feature in the stacks menu, which is called RMSD. We're going to activate that, and there we go. And we're going to select one of the proteins as an input, and then we'll select the other as a target. And I like to align the frames as well. So now when we run this, what you should see is that one protein will move to align with the other one, and you'll see this box created. And what that tells me is that I can now just grab them both and move them around with one hand. So you can see this did a pretty good job of overlaying the two ligands, and you can see that the hinge is overlaid. There's a little bit of a difference between these two loops here. This is the glycine loop that sits over the top of the ATP binding site. But globally, there's a lot of very close similarity between the two protein structures. But there's one really significant difference, and it's in the back here. And it's this coil region here, this alpha helix. So remember, the blue is our 2ITY structure. That's the gefitinib or ERESA structure bound to EGFR kinase. And then the yellow is that structure is the lapatinib structure bound to EGFR kinase. And I color-coded the ligand so you can see which ligand goes with which set of coils. So the larger ligand is actually pushing that coil back away from the ATP binding site. And what that does, and we saw how there was a sort of the induction of a larger pocket through the bigger ligand. It's partly through the displacement of that helix. That's the C helix. And this is what we would refer to as a C alpha helix out structure. And so this is in fact not a type 1 binding mode of the kinase inhibitor, but what we call a type 1.5 ligand binding mode. And it's actually an inactive form of the protein. So even though the Dfg motif was in, it's not a type 2 binding mode, which would have the Dfg out. It's a type 1.5 where the Dfg motif is pointed in, but the C helix is pushed out. And so that's a distinction that I think sometimes is overlooked, but is a different way of inducing an inactive conformation of the EGFR kinase or any kinase domain. And so the lapatinib structure does a nice job of pushing that C helix out. So that's sort of visually what it looks like. What are the implications of that? Are there implications of having this structure in it, or this kinase in an inactive conformation? So this is the web browser that we used earlier to download the first protein. And if you can see this, let me make it a little bigger. So this is a table in a publication about residence time and the kinetics of ligand binding. And you can see in this table here that gefitinib and lapatinib, the two ligands that we were just looked at, are listed in this table. So what is shown in this table? This is actually a series of binding constants and kinetic rate constants for these two ligands bound to the EGFR kinase. So the first thing to note is that if you look at their binding affinity, so we have a Ki and a KD here, they're pretty similar. They're both in the nanomolar range. In fact, gefitinib has an order of magnitude, roughly almost an order of magnitude higher Ki, but it's a slightly weaker KD. But they're both similar. And when you look at the on rates, they're also not too different. So 5 times 10 to the 6 molar per second for gefitinib, 1 times 10 to the 5 for lapatinib. But when we look at the off rates, there's almost 2 orders of magnitude difference. And so what that's telling us is that when we look at the kinetics of binding, lapatinib actually comes off the protein more slowly than gefitinib comes off the protein. And the consequence of that, if you just invert the off rate, is that you get a residence time, and you could see the 2 different residence times here. So for gefitinib, it's 5 minutes, but for lapatinib, the larger ligand, it's 354 minutes, or basically 6 hours. And when you look at the activity of these 2 proteins in cell-based assays, and you treat the cells with the inhibitor and then wash it out and look to see how quickly does the activity of the kinase recover, you can see that for gefitinib, it's about 2.6 days, but for lapatinib, it's well over 4 days, and in fact, only about 20% of the activity has been recovered after 4 days after washing out the ligand. And so there's a pharmacodynamic effect of the induced change in the conformation of the protein as a consequence of lapatinib binding. The induction of that inactive conformation has a consequence on the duration of the effect of the inhibitor pharmacodynamically, both in cells and also you see that in vivo. And so this is a very significant consequence of the induced conformational change in this protein from changing it from an active to an inactive conformation, and suggests perhaps that it's a good idea to bind other ligands, to design other ligands for kinases that can induce C-helix out inactive conformations of the protein if you're interested in inhibiting kinases for drug discovery. So that was really the story that I wanted to tell, and hopefully this has been useful and interesting.
Great. Yeah, thank you so much.
That was fantastic.
Very educational.
Yeah.
Oh, can I ask you a question?
Sure.
Hello. Yeah. So I guess there are structures of this kinase like without the ligands, right? So it's really that also proves that the binding changes the normal conformation of the protein.
The holo structures without any ligands bound generally are found to be in a more active state or conformation. Typically, of course, what you might see is a structure with an ATP or an ADP bound, which would be in the active conformation. But yeah, so I'm not aware. I wouldn't swear that there aren't any, but I'm not aware of any crystal structures of sort of C-helix out or type 2 binding modes with the DFG out that are consequent that don't have a ligand bound. Now, there's definitely a school of thought that the protein might be able to adopt those conformations independent of ligand binding, and that the ligands are simply stabilizing those conformations and selecting them from an ensemble of protein conformations, all of which exist in solution. And that's definitely a possibility, but I don't think that I'm certainly not aware, and a crystallographer may correct me, but I'm certainly not aware of any crystal structures of kinases where the inactive conformation is present without a ligand being bound. So that's why when we talk about these conformations, we tend to talk about them in the context of the ligand and the binding mode of the ligand. So we talk about a type 1, type 1.5, type 2 binding mode of the ligand, rather than talking about it as a state of the protein.
Got it.
And just to kind of add on a little bit, just so that it kind of makes sense, because then the question might be, well, if kinases are never able to be found in like an inactive state, does that mean that they're always active? And I just want to mention that there are lots of other parts of this protein where we are just looking at the kinase domain, whereas there's typically a number of regulatory domains that will block this area with a separate component of the protein. And so while we talked about the active and inactive state, that isn't merely a description of how the protein functions overall, but really describing, as you know, Peter was describing rather well, like the DFG and how it's able to facilitate the catalytic site and its involvement with the ATP molecule in any target protein.
Yeah, that's a really good point, John. And also phosphorylation of the kinase domains has an effect on the activation state and the kinase function. So yes, there are many other regulatory mechanisms for kinases. It's not all about just the ligand, but of course, as whilst we can't ignore the phosphorylation state and the regulatory factors, particularly when we're setting up assays, of course, as drug design scientists, we are frequently focused on the ligands.
All right, thank you so much, Peter. That was incredibly helpful and informative. And we hope that we can have you again in the near future. Thanks again. Thanks, everybody, for joining. And we'll see everybody next time. Bye-bye.
Yeah. Bye-bye.
All right, bye.